Welcome to the Deep Dive. Today, we're looking at how we understand life at the molecular level. It's been quite a journey, hasn't it?
It really has. I mean, for years, biological research sort of operated in stages.
Right. First, we had bulk RNA sequencing. That told us what genes were firing up across, say, a whole chunk of tissue. A good overview, but averaged out.
Totally average. You lost all the nuance. Then came the revolution. Single-cell RNA sequencing, up here, and a sec. Suddenly, we could see which specific cell types were doing what. Incredible clarity.
Incredible clarity, yes, but with a pretty major catch, wouldn't you say?
Oh, absolutely. A fundamental flaw, really. To get that single-cell detail, you had to basically, well, dissolve the tissue. You got the players, but you completely lost the playing field.
The architecture, the context.
Exactly. The native environment. And that location, that spatial context, you could argue it's the most critical piece of the puzzle in biology.
We often say disease is a neighborhood problem, don't we? It's not just a list of bad ingredients. It's about how they're arranged and interacting.
Precisely. So the next frontier, the place we really need to be, is spatial transcriptomics.
ST. And ST is the tech that finally lets us quantify RNA within an intact tissue slice. You see the gene expression patterns right where they happen.
It's genuinely transformative. But, like any rapidly evolving field, it brings complications. You've probably noticed this if you're working in the area. There's now, well, a real zoo of different ST platforms.
The zoo's a good word for it. You've got 10x GeoMx with Visium, their newer Visium HD, and the Xenium. Then there's NanoString's GeoMx and CosMx. It's a lot.
And they're not just minor variations, they're fundamentally different approaches. GeoMx, for example, looks at specific regions you draw, like a kind of mini bulk analysis.
Okay.
Whereas Xenium or CosMx are imaging-based, aiming for true single-cell resolution in situ.
Right, so it's like trying to compare data from, I don't know, a wide-angle survey camera versus a high-powered microscope focused on individual details.
Yeah.
The data formats, the resolution, what they actually output, is all over the place.
You've hit the nail on the head. This diversity, while powerful in itself, creates a huge fragmentation problem. Imagine you're a researcher.
Okay.
And you've got kidney data from GeoMx and maybe some brain tissue data from Visium. Just getting those data sets into a comparable format for analysis can take weeks. It's a massive headache.
So there's this critical gap, a real need for some kind of standardized way to handle it all, end to end.
Exactly. And that's where SpaceSequest, the focus of our deep dive today, comes into the picture.
The solution.
Well, it aims to be. It's a new computational pipeline specifically designed to be this central analysis hub. Tailored, comprehensive, and unified across those 5 major ST technologies we mentioned.
So what's its mission in a nutshell?
It's fourfold, really. 1, standardized quality control, getting rid of the junk data consistently. 2, customized analysis workflows that still feed into a unified framework. 3, automated cell type annotation, and crucially, deconvolution, figuring out the cell mix.
That sounds like the really tricky part.
It is. And 4, generating high-quality, standardized outputs that actually make it easier for people to compare results and collaborate. Basically, it wants to bring order to the spatial chaos.
Okay, before we dive into the mechanics of how SpaceSequest does all this, let's just pause for a second. Why is this spatial information so important? Why put in all this computational effort just to know the address of a gene, especially in something complex like a tumor or, say, Alzheimer's disease?
It matters so much because disease isn't uniform. Think about cancer or neurodegeneration. These are driven by very specific interactions between different cell types in particular locations. ST lets us see that local heterogeneity.
Right.
If you just grind up a tumor and do bulk sequencing, you average everything out. You completely miss that critical, say, interaction zone between immune cells flooding in and the cancer cells right at the edge, the invasive front. That's where the action is.
I remember seeing a study, I think it was on prostate cancer, using ST. It was striking. They didn't just map the tumor. They found these gradients of gene expression around it. It showed how the nearby normal tissue, the stroma, was fundamentally changing its tune right next to the aggressive cancer cells. That changes everything about how you might think about treatment or progression.
That's a perfect example. And it leads us nicely into the 3 sort of key biological insights ST really unlocks for researchers.
Okay, what are they?
First is just mapping the neighborhood. Cell type composition. ST lets us figure out what cell types are present and how they're organized. We can build these incredibly detailed atlases for complex organs, think heart, kidney, brain, showing the precise cellular architecture.
So, number 1 is the blueprint, the static map. What else? Does it tell us how these neighbors talk to each other?
Yes, that's the second crucial role, understanding cellular interactions. SD doesn't just show who's there, but who likes to sit next to whom. It reveals patterns of spatial covariation.
Like attracts like or specific pairings?
Exactly. For instance, in the mouse visual cortex, studies have shown that certain types of inhibitory neurons prefer to be near each other, suggesting they form functional units. Or in Alzheimer's, seeing microglia consistently clustering around amyloid plaques tells you something vital about the disease process. If two cell types are always found together, they're likely involved in some shared function or pathway.
Okay, so we have the map, and we have the preferred seating arrangements. What's the third role? Getting down to the actual messages being passed.
Precisely. Molecular communication. Especially with the sequencing-based methods that capture the whole transcriptome, we can go beyond just proximity. We can look for specific signals, ligand molecules produced by one cell type, and receptor molecules on an adjacent cell type that can receive that signal.
Mapping the actual conversation.
Right. You can identify these ligand receptor pairs between neighboring cells and start to map out the complex signaling networks. This is critical for understanding everything from how tissues develop in an embryo with signaling gradients to how tumors manipulate their local environment to help them grow and spread.
And this challenge, understanding the communication, brings us right back to those two main families of ST technology that SpaceSequest needs to handle. You mentioned sequencing-based. Visium is the main one there.
Visium, yes, and its high-definition successor, Visium HD. Also, GeoMx falls partly into this category. The core idea for Visium is a special slide, a surface coated with millions of tiny spots, each with a unique DNA barcode.
Okay.
You place your tissue section onto this slide. The RNA molecules from the cells diffuse down, get captured by the nearest spot, and are tagged with that spot's unique barcode. Then you sequence everything.
And because you know which barcode corresponds to which spot on the slide.
You know roughly where that RNA came from in the tissue. This gives you an unbiased look at the entire transcriptome across the tissue section. Fantastic for discovery for hypothesis generation.
But there's always a trade-off, isn't there? What's the catch with Visium's approach? Resolution, I assume? Those spots aren't single cells?
That's the main limitation. The standard Visium spots are 55 micrometers across. That's easily big enough to capture RNA from multiple cells. You might get signals from an astrocyte, a couple of neurons, maybe a microglial cell, all mixed together in one spot's data.
Right, so you don't get that pristine single-cell view directly from the measurement.
Not directly, no. Now contrast that with the other family, imaging-based ST. This includes platforms like Xenium and CosMx.
And how do they work?
They use techniques based on fluorescence in situ hybridization, or FH, or similar in situ sequencing approaches. Essentially, they use fluorescent probes that bind directly to specific RNA molecules inside the cells, within the intact tissue. Then they image it.
So you can literally see the glowing dots of RNA inside individual cells.
Exactly. This gives you true single-cell resolution, or even subcellular resolution in some cases. You know precisely which cell is expressing which gene from your target list. Fantastic for hypothesis testing.
But the trade-off here.
The trade-off is usually the number of genes you can look at. Instead of the whole transcriptome, these methods typically target a predefined panel, maybe a few hundred, maybe up to 1,000 or so genes, depending on the specific platform and experiment.
Okay, so sequencing-based gives you breadth, but mixed spots. Imaging-based gives you single-cell precision, but for fewer genes. If Visium gives you that smoothie spot with multiple cell types blended together, how does something like SpaceSequest figure out the ingredients? How does it unmix the signal?
That is the key computational challenge for grid-based data. The answer is a process called deconvolution.
Deconvolution, okay.
Since the spots give us a mixed signal, we need to use computational methods to figure out what went into the mix. We essentially need a reference.
A reference? Like what?
Typically, a detailed single-cell RNA-seq dataset from a similar tissue type. This cRNA-seq data gives us really clear signatures for each distinct cell type. What genes does an astrocyte typically express? What does a neuron express? And so on.
Got it. So you have the signature list.
Right. Then you take the mixed signal from a Visium spot and you use mathematical or statistical algorithms to basically say, okay, given this mixed gene expression profile in this spot, and knowing the typical signatures of astrocytes, neurons, and microglia from our reference, what's the most likely proportion of each of those cell types contributing to this spot's signal?
So it's like mathematically reconstructing the recipe for the smoothie.
That's a great analogy, yes.
Yeah.
It deconvolutes the mixed data to estimate the relative abundance of different cell types within each spatial spot. It turns that blurry mixed signal back into a much clearer map of the cellular neighborhood.
Okay, so SpaceSequest has this huge task. Take input from Visium grids, GeoMx ROIs, Xenium single cells, all these different formats, and somehow streamline the analysis. Let's get into the nuts and bolts. How is it built? What makes it this potential universal translator?
Well, the architecture is key. It's built using well-established, robust bioinformatics packages that many researchers already use, like Surat version 5 in the ANDATA format. That helps with adoption.
Familiar tools, okay.
But the crucial part is how it orchestrates the workflow. It standardizes those really complex steps, things like data loading, quality control, clustering to find spatial domains, and that deconvolution we just talked about, making them work consistently regardless of which of the 5 platforms the data came from.
Right. Let's talk inputs for a second, because this is practical stuff listeners might need. If I've just run a Visium experiment, what file exactly do I feed into SpaceSequest? Does it take the raw output?
Not quite the rawest output. You generally need to run the data through the manufacturer's initial processing software first. For 10x genomics platforms like Visium or Visium HD, that means running Space Ranger.
Okay, Space Ranger.
Space Ranger does the initial alignment and counting, and it generates what's called the UMI count matrix. UMI stands for Unique Molecular Identifier. This matrix is basically a big table telling you for each spot how many unique RNA molecules for each gene were detected. That's the primary input for SpaceSequest from Visium.
Got it. UMI count matrix from Space Ranger. What about GeoMx?
For GeoMx, the standard pipeline from NanoString generates something called DCC files, which stands for Digital Count Conversion. These files contain the normalized counts for the specific regions of interest, the ROIs that the researcher selected during the experiment.
So DCC files for GeoMx, and for the imaging-based ones, Xenium and CosMx.
Those tend to be more straightforward. Their outputs, often lists of transcripts with coordinates, can typically be loaded more directly, perhaps after some formatting, because they already represent single cells or transcripts.
Okay, so once the data is in the right starting format, SpaceSequest takes over the heavy lifting. Let's talk algorithms. Finding spatial patterns, or clustering, is fundamental. What tools does SpaceSequest use, especially for that mixed Visium data? Standard clustering.
It includes standard methods, sure, like using UMP for dimensionality reduction, then finding clusters, which works well for single-cell resolved data like Xenium or CosMx, or the binned Visium HD data. But for the standard Visium grids, it incorporates 2 really powerful specialized tools.
Okay.
The first is BayesSpace. This is a Bayesian statistical tool, which means it uses probabilities and can incorporate prior knowledge.
What kind of prior knowledge?
The knowledge that tissue structure isn't random. BayesSpace assumes that spots which are spatially close and have similar gene expression profiles are likely part of the same biological domain. It uses this spatial context to refine the clustering, essentially sharpening the boundaries between different tissue regions.
That sounds smart. Using the location itself to help interpret the gene data?
Exactly. And BayesSpace has another neat trick. It includes a method to try and computationally enhance the resolution. It can reallocate the gene counts from the original spots to sort of refined subspots, giving you a potentially finer-grained map.
Interesting. And the second tool?
The second is Spatial. This one uses graph convolutional networks, a type of deep learning that's really good at handling data with relationships, like spatial relationships.
Deep learning, okay. What makes Spatial special?
What's really clever about Spatial is that it integrates 3 layers of information simultaneously. The gene expression data itself, the spatial coordinates of the spots, and information extracted directly from the histology image, the actual picture of the stained tissue.
Wow, so it looks at the tissue's appearance, too.
Yes. It learns patterns that combine molecular state, location, and visual features from the H&E stain, for example. This multimodal approach can lead to really robust identification of spatial domains.
Okay, incorporating tools like BayesSpace and Spatial makes sense for Visium. But here's something I wonder. SpaceSequest bundles these, plus others we'll discuss like Tangram and Cell2location. How does a user navigate that? If you run the Visium workflow and get, say, slightly different cluster maps or cell type predictions from these different tools, which one do you trust? Does it become a bit of a black box?
That's a really important question, and SpaceSequest tackles it head-on, particularly in the Visium workflow. It's designed to leverage parallel computing think running multiple analyses at the same time.
Okay.
So it runs BayesSpace, Spatial, Tangram, and Cell2location simultaneously. The developers didn't just grab random algorithms. They specifically chose tools that performed really well in independent, rigorous benchmarking studies comparing different methods.
So they picked the current state-of-the-art based on evidence.
Exactly. The idea isn't necessarily to force the user to pick just one definitive result. It's more about providing multiple high-quality views of the data generated by top-performing algorithms. You get these different maps and can look for consensus or perhaps notice subtle differences that might point to interesting biology. It promotes exploration rather than prescribing a single answer.
That makes sense, providing a consensus view from the best tools. Okay, let's talk about those other two key algorithms, the ones specifically for deconvolution, that smoothie unmixing process for Visium data. You mentioned Tangram.
Yes, Tangram is one of the stars for deconvolution included in SpaceSequest. It's another deep learning framework. What Tangram excels at is aligning the reference single-cell RNA-seq data onto the spatial transcriptomics data.
Aligning them? How?
It learns a mapping that essentially projects the single cells from the reference dataset into the spatial locations of the ST data, creating these predicted single-cell resolution spatial maps. It can even integrate other information, like known anatomical regions, to guide the alignment. It's very sophisticated.
Okay, deep learning for alignment. And the second deconvolution tool is Cell2location.
Cell2location takes a different approach. It's a Bayesian statistical model. It's become very popular and performs exceptionally well.
How does the Bayesian approach work here?
It works in two main stages. First, it takes your reference, say RNA-seq data, and rigorously estimates the reference cell type signatures, basically, defining the characteristic gene expression profile for each cell type in your reference. It does this in a way that accounts for technical noise and variability.
Okay, it builds a robust signature key.
Exactly. Then, in the second stage, it takes the mRNA counts from each individual spot in your spatial data, and using that signature key it just learned, it statistically estimates the abundance of each reference cell type within that single spot.
So it gives you actual numbers, like this spot is likely 40% astrocytes, 50% neurons, 10% microglia.
Precisely. It provides a quantitative map showing the estimated number or proportion of each cell type at every spatial location. It's incredibly powerful for understanding the detailed cellular composition across the tissue.
Right. And finally, before we get to case studies, there's one more optional analysis SpaceSequest offers, mapping the molecular conversations using Spatial.
Yes, Spatial is focused on cell-cell communication. If you've done the deconvolution and know which cell types are where, Spatial takes it a step further.
Now,
It uses the spatial information, which cells are neighbors, and combines it with databases of known ligand receptor interactions. It builds a graph network model to predict active signaling pathways between adjacent cell types.
so it goes beyond just these cells are next to each other to this cell is likely sending this specific signal to its neighbor, which is likely activating this response.
Exactly. It models the whole chain, ligand receptor, downstream target genes. It tries to build that functional interaction map, turning simple adjacency into a predicted communication network. Super useful for understanding how different cell niches influence each other in development or disease. So we've talked about the tools inside SpaceSequest, but the real magic, perhaps, is how it packages these into specific, easy-to-use workflows for each platform. You don't need to be a coding expert for each individual tool.
How does that work in practice?
It uses simple command line calls to activate a specific workflow, like Visium or GeoMx. And the detailed parameters for your specific experiment, those are configured using standardized YAML files, which are relatively easy to read text files. So you set up your config, run one command, and it orchestrates the whole analysis for that platform.
Okay, let's walk through an example. The classic Visium workflow. The paper mentions testing it on a public data set from a mouse model of Alzheimer's disease. What does that workflow actually do with that data?
Right, so you start with the UMI count matrix generated by Space Ranger for your Alzheimer's mouse brain slices. The Visium workflow in SpaceSequest first performs some standard quality control, maybe removing spots with very few detected genes or molecules, spots that are likely just background noise.
Standard cleanup. Then what?
Then it really kicks into high gear. As we discussed, it uses parallel processing, typically setting up multiple jobs on a computing cluster, using something like Spatch to run BayesSpace, Cell2location, SpatGCN, and Tangram all at the same time on the QC data.
That parallel step is crucial, right? Otherwise, running these complex algorithms sequentially would take forever.
Absolutely. It makes these advanced analyses feasible in a reasonable time frame for researchers.
And what kind of results pop out at the end for that Alzheimer's model? What does the user actually see?
They get a whole suite of informative outputs and visualizations. For instance, the BayesSpace results would show spatially distinct clusters across the brain tissue, highlighting regions with similar overall gene expression patterns, maybe delineating the hippocampus from the cortex, or identifying specific layers.
Okay, broad tissue regions.
Then, crucially, the Cell2location output provides those quantitative cell type abundance maps we talked about. For the Alzheimer's model, you'd likely see maps showing where specific cell types like astrocytes, oligodendrocytes, or importantly, activated microglia are most abundant.
And you could see those microglia are clustering around the amyloid plaques characteristic of Alzheimer's.
Precisely. You could visually correlate the Cell2location map showing high microglia density with known plaque locations, or with regions identified by BayesSpace as having a disease-associated transcriptional signature. It provides that spatial context to the cellular pathology that you'd completely miss with non-spatial methods. It turns the raw counts into a visual story of the disease landscape.
That's powerful. Okay, let's switch gears completely to GeoMx. You emphasize this is different. It's a mini-bulk analysis of user-defined regions of interest, or ROIs.
Yes, the experimental approach is fundamentally different. With GeoMx, the researcher looks at the tissue under a microscope and physically shapes the ROIs around areas they find interesting, maybe outlining a tumor region versus adjacent normal tissue, or specific structures like glomeruli in the kidney. The instrument then measures the expression of a large panel of genes, often the whole transcriptome, within each of those drawn areas.
Well, you're not dealing with spots or single cells here, but bulk measurements from larger defined regions.
Exactly. Therefore, the main analytical goal is usually different too. It's less about deconvolution and more about straightforward differential expression, comparing the gene activity between different groups of ROIs.
So the SpaceSequest GeoMx workflow reflects that. No fancy deconvolution.
Correct. The GeoMx workflow is streamlined for this purpose. It takes the DCC count files from the GeoMx instrument, along with metadata describing the ROIs, like which ones are tumor and which are normal, and it runs a robust differential expression analysis comparing the groups the user specifies.
And the output, just a list of differentially expressed genes.
It generates the list, yes, but it does something quite clever with the output format. It produces results as standard comma-separated value, CSV files, but formatted in a way that's directly compatible with popular downstream interactive visualization tools, specifically mentioning CosMx.
Ah, CosMx, that's an R-SNE app, right? For exploring results.
Exactly. So SpaceSequest doesn't try to reinvent the wheel for visualization here. It does the core DE analysis and then hands off the results in a ready-to-use format. This bridges the gap. Researchers can easily upload these CSVs into CosMx or similar tools.
And then they can generate their own plots interactively.
Yes. They can immediately make PCA plots to see if the ROIs cluster by type, like seeing kidney glomerulus ROIs separate from tubule ROIs, generate volcano plots to quickly spot the most significant up or down regulated genes, or create sheet maps showing patterns of the top differentially expressed genes across all their ROIs. It makes the insights accessible and flexible.
That's a smart way to integrate do the heavy lifting computationally, then output to tools people already use for exploration. Okay, one more platform. The new kid on the block, Visium HD. Released in 2024, promising much higher resolution.
Yes, Visium HD is a big step up for the sequencing-based approach. It uses much smaller, essentially continuous capture areas, providing higher spatial resolution, potentially near single cell without gaps between spots.
So SpaceSequest is already equipped to handle this newer format.
It is. There's a dedicated Visium H workflow. It takes the output from the Visium HD version of Space Ranger, performs quality control now at the level of these smaller bins, does dimensionality reduction and clustering similar to how you'd handle single-cell data.
Okay, standard processing adapted for higher resolution.
But they also added a really neat specific function tailored for Visium HD experiments. It's called Get Visium HD Spot Call.
Get Visium HD Spot Call. What does that do?
Often when you run Visium HD, you also acquire a paired high-resolution immunofluorescence, IF, image of the same tissue section, maybe staining for cell nuclei, like DAPI, or a specific protein marker relevant to your study.
Right, so you have the gene expression map and a detailed visual map.
Exactly. This function, Get Visium HD Spot Call, cleverly extracts the average color intensity, the RGB values, from that paired IF image for each of the small spatial bins in the Visium HD data.
Why is that useful?
It allows the user to directly link the gene expression data back to visible features in the IF image. For instance, you could use the DAPI intensity to select only bins that clearly contain cell nuclei, filtering out empty areas. Or if you stone for a specific cancer marker, you could select only the bins that show high intensity for that marker protein and then look specifically at the gene expression within those visually confirmed positive areas. It provides a powerful way to integrate the molecular data with direct visual evidence from microscopy.
This has been incredibly illuminating. So wrapping up, SpaceSequest really emerges as this ambitious, much-needed effort to create a unified, user-friendly pipeline. It supports 5 major, quite different ST platforms.
Right, and it doesn't just handle the basics. It integrates some really sophisticated state-of-the-art algorithms, Tangram, Spatial, Cell2location, BayesSpace, to squeeze the maximum biological insight out of these complex data sets.
And it's all guided by the FAIR principles you mentioned: findable, accessible, interoperable, reusable.
Yes, that's a key philosophy behind it. By creating standardized workflows and outputs, especially for platforms that were previously hard to compare, it makes the data inherently more interoperable and reusable. It lowers the barrier for labs to work with multiple ST technologies and facilitates the kind of large-scale data sharing and meta-analysis the field really needs.
And importantly for you, the listener, if you're intrigued by this and maybe want to try it on your own spatial data, the tools are out there. The source code for SpaceSequest is available on GitHub.
And they provide a detailed tutorial using Bookdown, which walks you through the steps. It's designed to be accessible.
We've spent this time talking about how spatial transcriptomics generates these incredibly rich, detailed molecular maps of tissues. It's fascinating stuff.
Absolutely. But it's also crucial to remember, as we've seen today, that these maps are only as good, only as interpretable as the computational tools we use to analyze them, to integrate them, and to make sense of the patterns.
Which brings us to our final thought for you to mull over. We know ST technology isn't standing still. It's evolving incredibly fast. We're seeing constant improvements in resolution, moving from targeted panels to whole transcriptomes and imaging, bridging sequencing and imaging approaches.
The hardware keeps getting better and better.
So here's the question. As these technologies continue to advance and generate even richer, more diverse types of spatial data, what completely new biological questions we couldn't even ask before, become possible when we have pipelines like SpaceSequest that can seamlessly integrate all these disparate data streams? What happens when we can build one truly coherent, multimodal, potentially three-dimensional understanding of tissue structure, function, and disease? What doors does that unlock?